Thank you very much for the kind introduction. Also, thank you, Michele, for inviting me. It's really a pleasure. It's been amazing. Actually, a very short and pleasant set of talks here. Like, we're almost neighbors, and so it was really very convenient to come from the Erasmus. But I was not expecting the high quality of the talks, and we basically covered all the topics. We covered sequences of cells. We've had the beautiful 3D presentations. We had consciousness, autism, Alzheimer's, epilepsy, seizures, rewiring of the brain, hippocampus. So we're missing the cerebellum, and not any longer. So my little outline, I'm going to, reintroduce you to the cerebellum in case you have forgotten about it. So I'm going to also tell you about the funky complex spikes that appear there. I'm going to then make the point that anatomy is fundamental for 3D modeling and of the particular piece of the cerebellar system, the inferior olive. And then I'm going to use the model to make a couple of predictions and a couple of functional theoretical calculations. And perhaps dispelling a couple of cerebellar mysteries in the process. So, and the reason why I love to study the cerebellum, I mean, partly because obviously, I don't know if that's really the primary reason, but because mechanical engineering, like motor, as Sheraton would remind us, is the one thing that we can do in the world, the first thing, I mean, when we want to, unless we're locked in, of course. And evolutionarily very fundamental, but maybe the reason why I really like the cerebellum is because it's the funniest system in the brain, at least to my mind. And you see all these amazing functions. I mean, some of them very simple, like the vestibulo-ocular reflex displayed by a chicken and by a child, the optokinetic reflex of the squirrel, the visual-motor coordination of the cat, and the error correction on the flyability of humans. But despite the fact that we commit mistakes, we can also establish absolutely incredible motor patterns, and I urge you to pay attention to the fact that this happens in a sequence, and where everything is, every state is being calling the next state, and so this is also one of the things that the cerebellum is related or supposed to do, which is teaching us how to transition quickly between sets of motor behaviors, such as, for example, the production of syllables. When I'm machine gunning you with multiple sentences, I'm creating very complex motor patterns in my mouth and emitting air during that time, and so everything has to be coordinated nicely. In ataxia, for instance, we have the problem of slurred speech, which is a failure of pattern coordination in the muscles of the mouth. And the thing is, the cerebellum is also known, like historically, to do motor learning based on error. So how does it all come together? I made this movie with a couple of creative liberties not very long ago in Blender. We also had a little bit of 3D Blender animations. And it's kind of it's a little cartoon of how the signal is distributed in the cerebellar Purkinje cells, who are flat and interact with myriad of parallel fibers, 500,000 of them that actually run for 4 millimeters in the brain of the human and distribute the signal linearly. There's a reason for that, and probably, else nature probably wouldn't have come up with it. I mean, here I cloned the cells. They're really one cell reconstructed. They're not all equal, as you can imagine. And I here represent a parallel fiber volley, which is a very sharp signal that comes through the mossy fibers and where things enter in the cerebellum. Here you see that the cerebellum stands in the middle of everything. I mean, it's a highly neglected structure, but it interacts with many other structures. And my claim is partly because it is such a contextual organ, creating these functions in a sequence. It actually is, combining the past with the future in a very short time scale. I'm talking really, 30, 40, 50 milliseconds. And so in this 30, 40, 50 milliseconds, we're going from loops that are ascending up to the cortex through the thalamus and down to the spinal cord, at the same time that ascending to many midbrain centers, many of them autonomous, like respiration in yoga. It's fastigial nucleus is known to control also respiration under load. And the amazing geometry doesn't stop at the parallel fibers. I mean, it's a very particular way in which it unfolds, like the cortex unfolds like a pizza. The cerebellum, yeah, that's not a pizza. So I urge you to then look at these unfolded cerebella and imagine or distinguish the cerebellum from the cow and from the human. So the topic of my talk today is actually not so much the cerebellum itself, but the cerebellar system, and in that one particular nucleus, it's a nucleus that is in many organisms, like the inferior olive nucleus, which is part of the cerebellar system that has very particular dynamics and structure and function. So the inferior olive of the human, so that you have a picture in your head, is huge. And in fact, the parts of it that grew the most were actually the parts that are correlated with cognition, which is an interesting connection that I'm not going to make today. Some people have, I think, very persuasively argued that the autism can be based on cerebellar dysfunction. So the olive is here in the midbrain, is participating in a loop with the cerebellum, and I actually had it in the slide but didn't talk about it. And I'm going to come back to how this loop looks like, but ultimately it sends very powerful signals with the so-called climbing fiber onto the cerebellar Purkinje cell, with which it overlays. So if the Purkinje cell is flat, the climbing fiber climbs and overlay parts of the arbor, especially the smooth parts of the arbor, and gives a very powerful signal, the complex spike. And so this is really funny. I mean, this is the projection from a small set of inferior olive cells from the rat onto the cerebellum, and you can see that in the unfolded cerebellum it actually fits in a very thin strip, and then we're wondering. I mean, the Purkinje cells are flat, and there is a very tight arrangement as they're being mapped from the inferior olive. And if you look at the complex spike signals that are appearing in the cortex, say for example with beautiful 2-photon imaging done by a colleague of mine. Let's see, I hope that this, so let me just tell you quickly, you're looking at the Purkinje cells from above, and this is calcium imaging during awake and behaving, so the mouse is actually walking spontaneously on a disc. But you can see, these are basically complex spikes that are appearing in the Purkinje cell. There are two kinds of spikes in the Purkinje cell, the simple spikes and the complex spikes. Simple spikes are very fast, complex spikes comes about once a second, which is also kind of funny because very infrequent. Now, having gone through my introductory slides, I'm going to and you see here some features that I might relate to. Like it's funny. What's the order of these cells firing? I think that this is one of the biggest questions for us. And how do I combine this with anatomy? Like one of my scientific heroes is Valentino Breitenberg. Amazing Italian neuroanatomist, also a systems thinker, who I think had many insights about many brain pieces. And I'm delighted with this quote because he said, 25 years ago, I thought the cerebellum was the part of the brain most likely to be understood by direct translation of the anatomy into functional terms. Today, I'm still confident, even though schemes proposed in the meantime have proved to cover at best only very partial aspects of cerebellar function, and sometimes quite divorced from any realistic consideration of histology. He was actually making a point about cerebellar theory. And Marr and Albus theory, some people will know that. And he made that quote in 1983. It's quite a while later, and we actually haven't figured it out yet. I mean, our considerations that were as divorced from anatomy back in the day are kind of like the still ones that reign in the dogma. But and so the question that I have for today is basically how does the inferior olive work, but it's in this overarching question of how does the cerebellum work, and I hope that it will give you some pictures of how it could potentially work. It's modeling work. So it's computational modeling work. We had very few of this, and though we're doing calcium and electrophysiology that corroborates some of these results, I will not show any of that, not today at least. And so if the question is how does the inferior olive work, I mean, we work with a much simpler question, which is how does the model of the inferior olive work? So for that, I'm trying to, like, obviously, it's biophysically plausible, but it's way more tractable because first we can actually see big features of propagation and membrane potential transmission, current flows, never forgetting how it actually looks like. So this is, the point that I'm actually making here is an old nucleus. And on the left here, you see very particular curly cells for which the olive is famous. And you see that they actually, you know, obviously they're not floating in space. I mean, it's not a Golgi stain, and so they're packed. And they're talking to their neighbors. And one of the reasons why they would like to curl like that is to maximize local connectivity. So another important thing is the afferent arborizations fill space, not only two-dimensional but three-dimensional. You have two kinds of arborizations, excitatory and inhibitory, and I'll definitely have the time to cover both of them because you know that is the essence of one of the functional essences of the message. And the one thing that it does dynamically, I mean, like if that's the anatomy, you have these cells that are very close by connected. They're also connected heavily with gap junctions, connection 36 in the adult, connection 45 in the young. This is a very nice experiment by Lesnick and Nuss, 2004, in which they were showing basically, like on the top, you see voltage-sensitive dyes with a cell that has been injected with a dye, and you can see that it's talking to the cells close by. On the left, you see the optical signal from the whole slice, and it's a millimeter the scale, plus the oscillation that is on the membrane potential of an intracellular recorded. And then you can see, well, they're oscillating, and the cell oscillates in tandem with the optical signal. If you block the gap junctions on the bottom row, these oscillations, the global oscillations disappear, but the oscillation in the single cell is retained. And so the black line, but the optical signal flattens out. So the global oscillations depend on the gap junctions, but the cells oscillate all by themselves. We keep this. I now notice that I have a slide that doesn't belong here. So having seen what are some of the important anatomical features, the overlapping cells, and some of the important dynamical features, which is like group oscillations, we then note that the complex spikes that we record in the cerebellum are very, very enigmatic. I mean, they're like super powerful, as I mentioned. This is a little review of the complex spikes. So they have multiple waveforms in their spikes themselves. They're very weird because they're actually unreliable, low frequency and broad latency to stimulus, and I think there are good reasons for that, and we'll see that. They're very multimodal. You can create them with a variety of stimuli, and so the receptive fields are very broad. They can be very synchronous in patterns, but that is contingent. I mean, like it's actually also happening in time. As you saw in the calcium imaging, you had what appear to be sequences. Wait a second. Oh, yeah. And so the questions for me then today are, what are the potentialities of the tissue? So if it is an old piece of the brain, it's actually not necessarily learning a lot. I mean, we know a lot of quadrupeds that are popped into existence and they're already basically able to move around and to do many of the functions the cerebellum is associated with. And so the question, so these are the properties that it has. I have like one slide in the future. So what are the potentialities of the neural tissue? How does it respond to particular stimuli? And how can a system with components that oscillate stiffly? I didn't mention that the cells' intrinsic subthreshold oscillations are fairly stiff. Will underlie functional behavior such as walking on a treadmill in which you accelerate smoothly, right? I mean, you have this kind of smoothness in motor behavior, probably subserved also by very likely subserved by the inferior olive. How does it work? I mean, like how do you put these low cells that oscillate stiffly to make things move smoothly and increase frequency smoothly? And so I'm going to show you directly what the model looks like in a three-dimensional plot now. And I'll just give you very broad details of the model. I'm happy to talk at length about how it's actually cobbled up. But it's a couple of thousand cells that we have, and we try to be very strict about how the cells are connected locally. And we have gap junctions that are uniformly distributed. And here in this graph, you see basically a chunk of this model in which the transparency will be the membrane potential when it's very low. The hot colors are the membrane potential when it's high, and you notice that it's at the -20, so it's basically at spiking threshold. And so if you just let it be, it oscillates, and like in Linaz and Lesnick's observation, it just kind of, globally, so to speak. This is at the control of calcium low threshold ion channels and the gap junctions. Now, what do you do with this? I mean, it doesn't seem to, it is oscillating. We actually, been seeing these things also in vivo. But we actually don't know how it works, because usually the mouse is in the setup. It's a very hard place to record from, because it's in the center of the brain, but it subserves all that function that, motor function that we see. So what I decided to do is to look simply at what happens when it receives excitatory and inhibitory stimulus. I actually put, there's also plasticity here, but if you then give stimulus to one subset of the cells, and in a subset that you have these efferents that are enclosing a little volume and talking to local cells that are all connected, what does then the model do like? And so I do both excitatory and an inhibitory stimulus. Just for context, I want to give you the slide. It's showing that it's work by a friend of mine, Maruka Yossisadi, also in Yarum's lab in Jerusalem, where she shows for the first time that in the mouse there are motor cortical monosynaptic connections into the inferior olive of the mouse. So that's where it's getting its excitation from. It's all other sources. But on the PO there, which is the principal olive, it's one of the subnucleus, you see axons that are basically closing up a volume, and they come from a very small piece of the motor cortex. They're doing somatic connections. And so, this is the glutamatergic connections onto these cells. Now, if you, yes? Rotate. So on the left here is the unperturbed, the same thing, and on the right, what I'm going to do is, you see the time of the simulation. At 600 milliseconds, I'm going to give it a bump, like a 10 millisecond excitatory stimulus. And what I now did, I detached a subvolume of the cells. Basically, I changed the phase, or I changed the moment of the oscillation of these cells. And this is really funny, because I'm actually only giving the stimulus once, and is retaining this, although you have the gap junction. So eventually, they're going to collect themselves again. This is one of the questions: how long does it take? I mean, how long does it take? Does one take to forget the motor action one is in one moment? So it's dislodging a little volume, and the dislodging of the little volume here on the right, you see a raster plot of complex spikes produced by the model, where time runs down, and I have here 1,000 neurons of this model, where the volume that I detach. You see that now in the color map, the membrane potential, and you see that the volume that I talked to is basically has advanced. I mean, you can maybe see that here in those spikes, and here are many more spikes in the rebound. I can talk about this response is also very phase dependent, where the spikes will occur or not, which will then be a mechanism of selection of action. And so this was upon excitation, what I just showed you. It gets most of its inhibition, the inferior olive, from the cerebellum, and it has the opposite effect. I mean, it's a little bit natural, but there are a couple of details how these things vary, having to do with the properties of the GABAergic synapse and properties of how the neurotransmitter is delivered in the volume. But ultimately, this is the two directions where it can go. And now we can ask, depending on the phase, maybe that's the question you guys have in mind now. So if it's oscillating and you bump it, what happens like in different moments that you bump, and what happens with excitation and inhibition when you bump? For that, we use a little bit of phase analysis here, and in which we convert the oscillation into a phase. I presume, neuroscientists, many will understand this. We select arbitrarily one point to say that that's our zero in phase, and in fact you will see my mapping there. So in the bottom you have the membrane potential equivalent, and here on the top you have the going from minus pi to pi, the oscillations. That now you will see what happens in the phase of the two groups, the stimulated and non-stimulated group, as I bump it. And so here you have first the blue and the red cells are all together, and then after the stimulation, the group was detached. Now this is interesting. It has a little transit memory. On the bottom you can see the phase as it's retained over a longer period of time. And this is 1 second. It's even contingent whether it's relevant for the cerebellum to remember the whole second. But still, in principle, and this is a very robust finding in these models. We can change a number of properties and have very similar behavior. So I believe that being that simple is actually one of the core findings about, or predictions, which seem to be corroborated by some studies, about old nuclei. So I wanted you to think about the fact that, in the middle of the brain, most of the mechanics of it are actually fairly old, and it doesn't learn a lot, not like in the cortex, right? You don't need 4 or 5 years until you go breathing. And so many of these things have to be in place, sort of from the onset. Now, this is stimulation in one particular frequency. If we then summarize the stimulations from inhibition and from excitation as a function of phase, we then see that on the left here, upon inhibition, you have very interesting effects. For early phases, you have an advance. For the later phases, you have a little bit of a delay, whereas in excitation, you have a more linear response. And incrementally advancing as a function of where you bump. And so you can, in principle, smoothly control the differences, the phase differences between these detached groups. It's theory, but we can construct very quickly a connection to how this subserves smooth movement in the motor system, where we need, for example, to create very tight phase differences between agonists and antagonists. And so this is another way to summarize this data. Qualitative features abound, and I think that many of them are interpretable, and many of them give very direct predictions about what happens in the system. And in fact, we're observing many of these turning out to be true, and I'll not show you data today, probably don't have enough time. So the idea here was that you would get which one is inhibition, which one is excitation. Another way to summarize this is a little bit less confusing. Don't mind this. This is for me. Every line is a simulation, and I have to see everything happens in big parameter spaces. But here, so this is a phase response curve. So it basically informs what happens with the phase of the group as a function of the phase that I stimulate upon inhibition and excitation. So, and very simply here in the inhibition, you have during the phase, which is represented by the membrane potential single wave. What happens with the phase of the group as you stimulate there, so it's sort of linear. And with excitation, you have this big, abrupt bump in the middle. It's where the complex spike is produced. Now, this is also very funny. So the complex spike is selective for a certain phase. This is actually known from slice work. The model reproduces it. And it then tells us that we can play with inhibition and excitation, changing the relative phases of the fibers or the cells that are actually controlling muscle groups in the cerebellum. So the idea is that these olivary cells and these firings here are talking very directly, one with one Purkinje cell exclusively and creating those sequences that we see in the calcium imaging. And then one of the fun things you can do is so the next plot you will see voxels because the rendering with the smooth membrane potential interpolation are very costly. I just wanted to show you one of the things that you can do with it and how you can use this, what I believe is some sort of transient memory of the neuronal groups. And so this would be it. I stimulate now in a sequence once, and the substrate is able to keep that sequence for a while. This is a known property from many highly interconnected systems called excitable media. And there were all the hype in nature a couple of years ago because it reappears, and then nature seems to like to use these things very much. You have systems that are highly interconnected, where the local neighborhood is informing all the local neighbors, kind of broadly, and you have activity propagation, but you also have this memory. So in the cerebellum, as I mentioned earlier, you have this requirement of knowing very well where you've been in order to transform to go to the next pattern that you want to create, next motor pattern you want to create. There are many results in the literature that point to phase differences being fundamental in the cerebellum. This is only one example here. I'm not going to go into details, but ultimately, you can find in the cerebellum spontaneously cells that are not complex spikes, they're not synchronous. They're actually happening in a sequence, and they're keeping static phase differences even through a long time, which indicates that many of these processes are, physical propagation will actually explain these kinds of effects. And so this is a short interim conclusion. I was just saying that in a 3D setting, simulation 2D I/O can detach dynamical cell clusters, not anatomical. It's important for cerebellar scientists, we can discuss, mechanism for a transient spatial temporal memory. And you can, it's not only one kind of pattern that it can reproduce, it could also in principle compute interpolations between the groups, which is, like you want to smoothen out an action, you might want to compute on the phase differences. And it's a mechanism for a smooth control. Now, the funny thing is, these two signals interact in the brain. The excitatory and inhibitory stimulus arrive at the inferior olive simultaneously. Or not simultaneously necessarily. They arrive at the inferior olive from different sources. It has been related in former theories to basically be a comparator in the brain. And a temporal comparator, no less, and would produce then complex spikes when there's an error, where there's a prediction error. The cat is walking out of the table, there's whoop, 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 where's the floor? I mean, during that time, he basically is producing a lot of complex spikes. One might like to think that those are predictive errors, and they are known to be related to complex spikes in locomotion. People have done these experiments, and and so we know where complex spikes appear with respect to surprise, but it's a motor surprise. If I now calculate the probability of complex spike appearance as a function of interacting streams, input is converging onto the inferior olive. So this is the last point I'm going to make. So the input is converging onto a system that is already having intrinsic dynamics. That's like the brain. It's already oscillating, and then you have like input streams, and they converge onto patches, and we know that the anatomy is very close by. So here, I'm just going to show you a short example of what happens if you have excitation inhibition, and in the other condition, then reversed inhibition and excitation. I'm particularly happy with these results because they explain a very long-standing paradox in cerebellar science about the creation of complex spikes. But here, on the left, it's the other way around, eh? So here, if I inhibit and excite, you produce a lot of spikes. You can see that the white axes are a little bit different. Here, this is 40 millivolts and 20, so this is basically 40. Because the complex spikes appear, and they also appear in an order. You saw, this actually is a particularly crystalline system, so that you can really visualize this. It doesn't need to be this way, but in any case, so this propagation then creates spikes on a sequence if you inhibit and then excite. It's kind of a little bit counterintuitive. The physical explanation is simple. You increase the driving force by hyperpolarizing the cell a little bit more, and then you let it be. When you excite, you're surfing on that spring effect of the driving force from the hyperpolarized cell. In the reciprocal case, you have the excitation first, inhibition second. Which thwarts the spikes. And so this is actually really funny. I mean, so you could imagine that if excitation from the cortex precedes inhibition from the cerebellum, everything is fine. You don't need to produce any surprise complex spikes. If you do it the other way around, and there are like latencies that play with this role. I mean, we know the latencies from these guys. Then you produce the spikes, and this is the final idea here in this talk. When we're talking about the parallel fiber stimulation that is coming with multimodal stimulus to these Purkinje cells, and you're creating sequences between the different olivary cells that are mapping to the Purkinje cells, we have a mechanism of temporal selection of coordinated stimuli. Thinking about any kind of learning you want here, supervised by the calamine fibers or unsupervised, this is a mechanism that will create temporal orders into how synapses are reinforced. I'm not going to go into details into that idea, and I'm going to skip the video. This is my message today is the perceptron being an analogy for the Purkinje cell. I don't know if many of you are computational minded or not. The perceptron was one of the first ideas of what the neuron should do. But people stared at the neuron and said, "What does it do? What does it do?" And so, obviously, it does pattern recognition. Purkinje cell looks a little bit like a pattern recognizer. I'm not suggesting it is one, but it does look like one. And so these are the functional answers that I had. Just to summarize once again, what are the potentialities of the neural tissue? It can detach the subvolumes upon stimulation. How does it respond to stimulus in a both phase and polarity dependent way? How can a system with components that oscillate stiffly subserve flexible rhythmic modulations? You can produce sequences and read these sequences out according to the synergies of the motor system. How does the system produce smooth modulations by creating phase differences between the groups? How does it learn by creating Purkinje cell neuronal sequences that are then coordinating the contextual environment? I love this guy, Arthur Winfrey. Thought a lot. I mean, maybe one of the fathers of, or one of the people that brought oscillations to our attention, and complex systems particularly. And says, I avoid mathematical equations because the deeper motivation. He's the guy with circadian rhythms. Numerical exactitude is alien to the diversity of organic evolution. And pretense of exactitude often obscures the qualitative essentials. This is my take-home message, for myself. It's I love to quantify things very precisely, but sometimes, the scale where things matter is our crucial bit here. This is the acknowledgements that I had. Lovely collaborators, Jorn Negrello, who actually coded the first single-cell model. I think it's the best I/O model out there. I just did the network. Piotr Sokol, Elisabetta Javaroni, master students, and collaborators in Chris's lab, and great lab mates. Dank u. Merci.
Great. Thank you very much for this nice talk. Are there questions?
No electrophysiology, right?
Very nice talk, very creative, I think. But my question relates to how well are these neurons known in terms of ion channel topography, and in particular the position of the T channels versus the glutamate and GABA inputs?
Lovely question. So the topology, not very well known. Okay, so that's the essence of the question. But people have been looking a lot at oscillatory and non-oscillatory cells as they relate to the different subnuclei of the olive, which also relate to the different zones. And so people were searching for the kind of obvious correlates, like the curly and the straight cells. Do they have different calcium T expressions? The oscillatory and non-oscillatory cells, do they have different calcium T expressions? Results are confounded. We have different labs talking about different things, but in general it seems that at least with respect to the calcium T, they are close to, they're somatic primarily, and they do not particularly correlate with, they correlate with the oscillatory amplitude very much. They do not correlate with morphology of the cells. And in different subnuclei, calcium T are expressed in different amounts. So if you look at the medial accessory olive, which is probably related to torso, like there's less endemic oscillations, and the system acts a little bit more like a dampened oscillator, and so the cells reflect that. And I didn't talk at all about the glomeruli, which are really interesting because the gap junctions, they are really very localized and controlled by AMPA receptors at the glomeruli and GABA at the glomeruli too. And so that will also create plasticity in the gap junctions. But I haven't mentioned that. Yeah.
Other questions?
Where is this 1 second long memory timescale coming from? I'm thinking of the cardiac myocytes are able to synchronize quickly, very quickly. Why is not this synchronization is taking longer?
So the synchronization will be a function, or how long the synchronization is a function of the gap leak. So how these cells connect to each other. And so if they're out of phase and they have a lot of connection between them, you will have a re-equalization of it. Having said that, so it's probably a tunable parameter depending on what is the functional output of that particular motor behavior that you have. And so how long does it have to be? How long is a jump, right? So what is the fastest, or maybe in dancing, I mean, how long should you remember where you are? The transients in the body should be short. So I think that the fact that that in my model it remembers for very long, it's actually not fundamentally relevant. I think that it's way more important that it should remember effectively to a very short time window where actually motion happens. And so 50, 100 milliseconds, 150, very important. Phase differences, very important. Alignment when you're having complex coordinated tasks and you're training for something like, say, juggling, and it's like ball goes from here to here, your eyes are going from here to here, your hands going from here to here, you're establishing sequences. And so these sequences have to, as much as possible, be represented in the oscillatory ensemble differences. And I think that the length of the transient desynchronization then is less relevant as a oscillatory memory as it is relevant as a way to imprint the current state of the system in terms of its synergistic components, like agonists, antagonists. Do I answer the question? Okay.
Other questions? No more questions? Okay. Thank you once more.
Thank you. Thank you so much.
So I think you all just need a few more minutes of patience before we will announce the winners of the big contest.